How does the brain communicate within itself? The brain is generally responsible for everything that makes up the human experience. Thinking, talking, laughing, moving, eating, breathing, sleeping, dreaming, all of these things are controlled by the brain and much more. But what is the process that your brain uses to actually send signals within neural circuits and create these experiences? Today, I'm going to discuss the overall process of neurotransmission and how different neural circuits are responsible for creating these different aspects of your human experience. I'll first start out by outlining the steps of neurotransmission. Neurotransmission is the process by which two neurons, or cells within the brain, communicate messages to one another. Neurotransmission is made up of both chemical and electrical processes. Basically, your brain uses a combination of both chemicals and resulting electrical signals in order to communicate. So the first step of neurotransmission is neurotransmitters binding to dendrites on the presynaptic neuron. I'm going to go ahead and break down that statement a little bit. First, neurotransmitters are chemical molecules that your brain uses to send signals to different neurons and create some sort of an action within that neuron that receives the neurotransmitter. So basically, all there is to remember for now is that neurotransmitters are chemicals within the brain that are used to send messages. There are different types of neurotransmitters, and we'll cover a little bit of that later on in the lecture. The next part of that statement I want to break down is the word dendrite. Dendrites are these spiny extensions here on the actual cell body of the neuron, and what these dendrites are responsible for is receiving messages. So dendrites are going to be the site on the neuron where these neurotransmitters are going to bind in order to create some sort of a resulting action in this neuron. The next word I'm going to explain is presynaptic. This is called the presynaptic neuron because this neuron is ultimately going to send a message to this receiving neuron, which is called the postsynaptic neuron. The next step in neurotransmission is this resulting electrical signal. So the chemical binding of the neurotransmitter to the dendrites on the neuron is going to trigger an electrical response called an action potential. This action potential or electrical signal is going to move along this extension from the neuron cell body. This extension right here, where the electrical signal travels, is called the axon. Then, once this electrical signal reaches the end of the axon, what happens is yet another process of chemical signaling. Essentially, this electric action potential is going to trigger the release of other neurotransmitters, and these neurotransmitters are going to be released into the synapse. The synapse is the gap between the presynaptic neuron and the postsynaptic neuron, and that's where these chemical signals are going to be released so that they can move across the synapse and then ultimately bind to the dendrites on the postsynaptic neuron. Then, once those released neurotransmitters bind to the dendrites on the receiving or postsynaptic neuron, the process is going to start all over again, and then another action potential will be generated if that chemical signal is ultimately strong enough to generate it. So that was just an overview on neurotransmission, but now I'm going to go ahead and dive a little bit deeper into what an action potential actually is and how it occurs. The first thing that I want to explain about an action potential is that it's ultimately an all or nothing process. So what that means is you're not going to have a minor or weak or strong action potential. There's no such thing as gradations with action potentials. Either you're going to have no electrical signal or you're going to have the maximum entire electrical signal. It's all or nothing. And so let's say that across the cell membrane of the neuron, there's an electric potential of negative 120 millivolts. By the way, that electric potential would be generated by a difference or a gradient in the concentration of ions within the cell versus outside the cell. And so that's what creates this electric potential or electric difference. And so let's say that initially you have an electric potential of negative 120 millivolts across the cell membrane of the neuron. But in order to trigger an action potential, you would need a higher electric potential of negative 70 millivolts. And negative 70 millivolts then would be called the threshold potential that needs to be met in order for an action potential to be triggered. And so in order to bring the electric potential up from negative 120 millivolts to negative 70 millivolts, well, that's where the chemical signals come in. Let's say that you have a excitatory signal or an excitatory neurotransmitter. Well, that's going to decrease this electrical potential difference across the cell membrane and bring you closer to the actual triggering of an action potential. But then let's say that the neuron receives a negative signal, and that's going to bring it further away from the threshold that needs to be met for an action potential to be triggered. And so those two signals will be competing with one another. But then let's say the neuron receives more excitatory signals and it gets a little bit closer to the action potential threshold. And then there's a negative signal that brings it down again, but then there's enough of a positive signal received that you ultimately get to that negative 70 millivolt threshold. Well, what happens then, once you reach that threshold, that is going to be an action potential. Because of this all or nothing process, it doesn't matter what pathway you took to get from your original negative 120 millivolts to the negative 70 millivolts. Once you reach that threshold, you're going to get an action potential. And so this is just sort of a conceptual way to understand how an action potential works in the brain. Now I'm going to go ahead and explain it from a little bit more of a mathematical perspective. Essentially, what we just saw is the summation of chemical signals in order to bring the electric potential across the cell membrane from its starting point to the threshold point needed to generate an action potential. And so what I want to conceptually explain here is that let's say you start with your initial electrical potential, and then some sum of excitatory and inhibitory neurotransmitters will bring you to that threshold electrical potential, your V final, so to speak. So this is just sort of an overview of the way that neurotransmission will occur. Now I want to dive a little bit deeper into what I actually mean by positive signals or excitatory signals and negative signals or inhibitory signaling. Well, you saw in the graph that I showed earlier that there are going to be some chemical signals that will bring you closer to an action potential, and there are going to be some chemical signals that will bring the neuron further from an action potential. And so the defining feature of the effect that a neurotransmitter has on the neuron is going to be whether or not it brings it closer to that action potential. And so those signals that are going to be positive and bring the neuron closer to an action potential are doing something called depolarizing the neuron. And what that means is they're creating less of a difference between the inside of the neuron and the outside of the neuron. Essentially, there's less polarization across the cell membrane because there's less of a difference in the ionic makeup of the inside of the neuron and the outside of the neuron, and so there's less of that electrical potential. Meanwhile, chemical signals that will bring the neuron further away from an action potential are going to hyperpolarize the neuron because there's going to be more of a difference between the inside of the neuron and the outside of the neuron ionically. I'll now go ahead and explain some of the main mechanisms behind depolarization. As a reminder, depolarization is the process by which chemical signals in the brain can bring a neuron closer to firing an action potential. So this isn't going to be an exhaustive list of the way that depolarization can occur, but I do want to provide a more foundational introduction to some of the different ion channels that are typically responsible for depolarization. So first you have your calcium and sodium ion channels, and the reason that I group those together is because they are going to be responsible for shuttling positively charged ions, and then you also have chloride ion channels, which are going to bring about depolarization through movement of the negatively charged chloride ions. So let's say that in your neuron, you start off with an internal environment that's much more negative than the external environment. And so, because there's such a big difference in electric potential across the neuron cell membrane, you're going to be pretty far away from firing an action potential. So, if there were excitatory signals that were going to bring the neuron closer to an action potential, how would that occur? Well, in the case of your positively charged ion channels, your calcium ion channels and your sodium ion channels, you would have positively charged ions flowing into the cell and thus making the inside of the cell less negative and decreasing the electric potential across the cell membrane that way. Or with your chloride ion channels, you'll have negatively charged ions flowing out, making the outside more negative, and the result is the same. You decrease the electric potential across the neuron cell membrane. And so those are some of the main mechanisms behind depolarization. Next, I'll go ahead and explain hyperpolarization, and for this there are two main mechanisms I want to point out. So in this case, let's say that your neuron is very close to firing an action potential, but for whatever reason, there has to be inhibition of that action potential, and so inhibitory neurotransmitters might bind to the neuron, and there could be, as an example here, two cases. One, you have positively charged ions flowing out of the neuron, and so in that case, that'll make the inside more negative and the outside will be, relatively speaking, more positive, and so you increase the electric potential difference across the cell membrane. And then if you have a chloride ion channel that is performing hyperpolarization, then you would have negative ions flowing in. Once again, the result is the same. The inside of the neuron is going to become more negative compared to its outside external environment. And so as a result, the neuron will be drawn further away from the threshold potential required for an action potential. Now I'm going to go ahead and explain some of the different types of neurotransmitter receptors. So what we were discussing just now are the ion channels that are responsible for actually creating the electric potential difference needed for an action potential. What I'm going to talk about now are the chemical receptors on the surfaces of dendrites that are going to actually receive the neurotransmitters and then allow for an electrical response. So there are going to be two types of dendrite neurotransmitter receptors that I'm going to cover. First are ionotropic receptors, and second are metabotropic receptors. And so there are different components of neurotransmitter reception. The first are neurotransmitter binding, so the actual chemical, the neurotransmitter molecule, has to be able to bind to the receptor itself. But then the second component downstream of that is that once the chemical binds, it has to initiate some sort of an electrical ionic effect, meaning that the neuron will either become further polarized or it might become depolarized. And so you might ask, what's the difference between ionotropic and metabotropic receptors? And the answer is that the way in which the chemical binding is linked to the downstream electrical effect is going to be different. And I'm going to go more into depth into what I mean by that now. So I'm going to start off with explaining ionotropic receptors. Ionotropic receptors can be thought of as dual function because they have two components to them. They have a chemical binding site where the actual neurotransmitter binds, and then they have in them an ion channel as well. So let's say here that you have a chemical binding site on the surface of the ionotropic receptor, and that's going to fit with a lock and key mechanism to the neurotransmitter molecule. But then here you also have the ion channel that ions can flow through once the neurotransmitter binds to the surface of the ionotropic receptor. So let's say you have your neurotransmitter molecules, they bind, then immediately the actual ion channel will open up, and ions will be able to flow through the ionotropic receptor, and they might be flowing inside the neuron or outside the neuron, but in any case, within this one receptor, you have the dual function of chemical binding of a neurotransmitter, as well as the creation of a change in the electric potential across the neuron cell membrane. And so the same receptor that will bind neurotransmitters will also be able to affect the likelihood of an action potential firing. This particular type of receptor is going to be important when you need a fast neural circuit response. So because there is this dual function, you can get changes in the electric potential and the signaling across a neural circuit very, very quickly. And so some examples of when you would have ionotropic receptors, let's say that you are in the middle of a very stressful situation. Maybe, for example, you hear a car backfire and there's this sudden loud sound and you get very stressed and your initial response is to run away from the loud noise. Well, then ionotropic receptors in your brain would be responsible for creating that immediate fear response, as well as helping you go into the stress phase or the fight or flight response so that you can immediately have this change in behavior and escape that stressful situation. So that's just an example of when you would want a fast neural circuit response. Now I'm going to go ahead and cover metabotropic receptors. So metabotropic receptors are unlike ionotropic receptors in that they don't have both the chemical binding and electrical ion function. So what do I mean by that? Well, typically with metabotropic receptors, you will have the initial receptor that has the chemical binding site. And so, just like before, the neurotransmitter binds to the chemical binding site. But then, in order to create an actual change in behavior of the neuron, the metabotropic receptor will not just act like an ion channel and let ions through. One of two things tends to happen upon the neurotransmitter binding. The G protein at the base of the metabotropic receptor will be activated, and then either a downstream or nearby separate ion channel will be opened, and ions will flow through, or some internal enzyme cascade is going to cause cellular gene expression changes. And so that'll result in a gradual neural circuit response. Because in this case, there's not a conservation of chemical binding site and ion channel function within just one protein. In this case, you're going to have downstream effects of some downstream ion channel opening or cellular gene expression changing as a result of neurotransmitter binding. So with the gradual neural circuit response, this type of receptor might be more common in neural circuits that are responsible for learning, which is a long-term process, or memory consolidation, just to give some examples. Okay, now I'm going to go ahead and cover different classes of neurotransmitters. So there are many different types of neurotransmitters in the brain that are going to be responsible for performing different functions as needed in different circumstances in your life. And so this isn't an exhaustive list, but I am going to cover some of the main classes that are typically seen in neurotransmitters. So first you have your soluble gases, and soluble gases are going to work by essentially dissolving in the bloodstream, and they're going to diffuse between the presynaptic neuron and the postsynaptic neuron. So, like I said, these move by diffusion in the blood, and oftentimes they're going to be useful for protecting against neural inflammation and also increasing mood control. So those are just some of the common functions of soluble gas neurotransmitters, and some examples are going to be nitrous oxide, hydrogen sulfide, and carbon monoxide. Okay, the next class of neurotransmitter are going to be amino acids. I went ahead and drew out the general structure of an amino acid. Typically, you have a central carbon attached to a hydrogen, an amine group or NH2, and then a carboxylic acid. And then on the side, you're going to have some sort of a side chain that's going to vary between different amino acids. That's not particularly important to memorize for the purpose of this lecture, but might be nice to know in case you're interested in the chemistry of neurotransmitters. So these particular types of neurotransmitters are typically responsible for neuromodulation, which is the more gradual process of changing the activity of a circuit, and it's responsible for fine-tuning the activity of a circuit. And then also, because they're amino acids and their small building block molecules, they're going to be responsible also for the participating or being part of the early synthesis of other neurotransmitters. So these are pretty useful building blocks then to build other types of neurotransmitters that we'll discuss a little later. So some examples of this are glutamate, which is the most prevalent excitatory neurotransmitter in the brain, glycine, and GABA, which is an inhibitory neurotransmitter. So these are pretty versatile in terms of their uses in the brain. Next, after amino acids, we have neuropeptides. And so peptides are essentially chains of amino acids. And so I chose to depict that here with a bunch of amino acids linked together. And so neuropeptides are going to be also long-term neuromodulators, and they're also responsible for a lot of maintenance of homeostasis, as well as monitoring basic states of the body, like the amount of pain you're in, or the amount of hunger you have, or the amount of thirst that you feel. So neuropeptides are going to be very important for maintaining your body on a day-to-day basis and also impacting the long-term activity of neural circuits. So some examples are going to be enkephalins and neuropeptide Y. The next class of neurotransmitter that I'd like to cover are monoamines. And for those of you interested in psychiatry, these are going to be particularly important. They're largely involved in mood regulation, reward system functions, executive functions like planning, long-term decision-making, and social interaction, and also sleep regulation. So their general chemical structure is that you will have some sort of a 6-carbon ring, and then you're going to have an amine as well that's attached somehow to that 6-carbon ring. So a lot of variations on this structure, but that's just typically the core principal structure of a monoamine. Some examples are going to be serotonin, which is famously involved in mood regulation, among other things, norepinephrine, and dopamine. And dopamine is famously involved in the reward system and the pleasure response. Okay, the last class of neurotransmitter that I'll cover is acetylcholine. And acetylcholine is really one neurotransmitter, but it's hugely important for voluntary movement, focus, autonomic system functions, like, for example, the movement of the muscles in your intestines for food digestion, as well as memory consolidation. Thank you so much for listening to this lecture. I hope that you were able to learn a lot about neurotransmission, and I'm excited to see you guys in the next lecture.